Hey guys, so we're talking cytokines, chemokines, chemotaxis. Let's get into it this week. We ended last time talking about T cell activation. So T cells need their T cell receptor plus a secondary receptor like CD28 to bind to an antigen presenting cell and receive those activation signals. Okay, that's pretty clear. But T cells can do a lot of different things, right? They could be T helper 1s, T helper 2s, Tregs, T17s. There's tons of subsets. And so our cytokines that get secreted are actually going to guide the immune response. And to guide the immune response, you have to tell your T cells to do something specific. The chart that we've seen before, a really nice one, is between T helper 1s and T helper 2s. And you can see all of these things are guided by specific cytokines. And we're going to go through all of the classes of cytokines that exist. And you can see some on this chart. We've got interleukins here. We've got some interferon here. And there's other ones that we're going to be talking about. But the idea is really, really important that a cytokine is just something that's going to get secreted that's going to guide some cell to do something. Well, how does that happen? Well, that's a good question. So let's talk about cytokines in general. So cytokines are signaling peptides. You can use the term growth factor. Those two terms used to be different, but now we can kind of associate them together. So they're used interchangeably in a lot of different fields, though it kind of depends on what you're talking about. Usually we associate cytokines with the immune system. That's where we discovered a lot of them, but it's not exclusive. There's tons of cytokines secreted by all kinds of cells. But those cells are going to secrete something when they receive a stimulus, right? So a cell receives some kind of stimulus. It could be a viral infection. It could be another hormone. It could be a cytokine. Who knows? And then it changes its transcription factors, starts secreting something, and then that goes binds to another receptor on a target cell. The concentrations of hormones versus cytokines is one way that we can sort of separate them out. So like a hormone coming out of like your pituitary gland would maybe be in the nanomolar range, whereas a cytokine would generally be sitting at a picomolar range. But it can fluctuate much more greatly. So like a hormone might jump up 10%, but a cytokine might jump up 1,000%. And so that can really have a big drastic effect. But it kind of makes sense. Your cytokines are generally being activated in the presence of like an infection. And so we only really need them to ramp up and cause whatever actions are going to cause when we actually have an infection around. One way to distinguish cytokines is to show how they are transmitted. So they can be autocrine, which they're acting on the exact same cell that they came out of, or they could be paracrine, which would be like acting on just nearby cells. And then you've got your endocrine action, which is how we would normally associate most hormones, where it gets into the circulatory system and can really change a lot of things. So if we actually look at our cytokines, it depends on how you're kind of parsing them out. But there's between 5 or 6 different classes or families of cytokines. I'm sticking with 6 here, but a lot of these there's some crossover between these, especially in the hematopoietins. But we're just going to go through each of these and just kind of address what each one does. And so let's talk about interferons first. So interferons were first discovered in 1957. They were the first of these cells discovered because they were the ones that were having the most direct effect on cells. So basically what they were doing was they had cell culture, and they would infect that cell culture with some virus. And then when they tried to add another virus in to sort of have like another infection happen, those cells were able to resist that virus. And what they've discovered was that these cells are secreting what we now know are interferon alpha and interferon beta. And what they were doing is that triggers a set of signaling processes in other nearby cells to warn them that there's a virus nearby, which I'll talk about on the next slide. But basically, the triggering of it actually makes perfect sense. It's just like all the other kind of signaling pathways that we're going to encounter. And so I'm not going to go ad nauseum, but just to hit this one right off the bat, I mean, we have the binding of our receptor, it dimerizes, and then it triggers phosphorylation. And then eventually you can phosphorylate what will become a transcription factor that then goes into the nucleus, binds to, your region that you need to amplify, and starts amplifying it. And so with interferons, you can see what we're generally getting. So let's say your epithelial cell, like in your lungs or something, gets infected, and it starts secreting interferons, and it warns nearby cells, and then that produces an antiviral state inside of them, and then that can go on and trigger other things too. Now, interferon doesn't have to come from those virally infected cells. It could come from one of your guards, like your antigen-presenting cells that's looking for infection, and they might say, oh my goodness, I found a piece of a virus. We need to secrete interferon to warn other nearby cells that, hey, something's going on. And so that could actually have an endocrine effect on your whole body. Interferons are what are making you kind of feel yucky when you have the flu. And so like you get malaise, you get tired, and that's generally interferon that's causing that, because it's triggering these special states in your cells. Your cells are almost becoming resting is too strong of a word because that's a specific term, but like your whole body is just kind of preparing itself and steeling itself against an infection. So I found a nice chart here that actually talks about what's happening inside of a cell, because generally interferons, especially interferon gamma, but also interferon alpha and beta, which this is the list for interferon alpha and beta. Those things are designed to trigger an antiviral state. And an antiviral state is just when your cell is able to resist a virus. And so what are the different ways that we can resist viruses? Well, there's a ton of different ways. But generally, what are your viruses? Well, you have viruses that are RNA, so we can destroy the RNA. So you can block RNA translation. You could degrade your RNA using RNases. You can directly go after like double-stranded RNA because that's really weird. So adenosine deaminase, that would be able to destroy specific nucleotides. You could ramp up the presence of specific reactive oxygen species like nitric oxide or things like that. This isn't a comprehensive list. One that actually makes perfect sense too is interferon can trigger the production of more MHC class 1, which MHC class 1's job, as we've learned already in this class, is to present antigens that are inside the cell on the surface. And so if you are doing that, then you would hopefully present a viral antigen, and then your cell could be destroyed before the virus can actually escape. Because we discovered interferons so long ago in like the 50s, they've been studied probably more than most cytokines. And so the effects of interferons are numerous, to say the least. In this case here, we've got interferon gamma here, and that's the one that more gets secreted by your antigen-presenting cells. And so that's the one that they're going to trigger, as opposed to alpha and beta. Those are generally the ones that are getting secreted by infected cells. So like, oh my gosh, I have a virus inside of me. And interferon gamma would be like, okay, my antigen-presenting cell bound to a viral antigen, and we recognize that viral antigen not necessarily just with our MHC class 2, but also with our pattern recognition receptors. That's like your toll-like receptors, which we'll talk about at a later time. Something else to think about is that viruses and whatever they're infecting, so in this case humans, we have been co-evolving with viruses forever, and so we have evolved ways to fight off viruses, interferon being one of those, and so viruses have evolved ways to block the interferon pathway because the more successful viruses might be able to do that. And so you can see just a few examples on this chart here. Anything in red is a specific virus blockage of a specific pathway that's triggered by interferon gamma. So normally it's supposed to be triggering these like gamma associated genes. So for one example, you need to use your proteasome to cut up any proteins that are inside the cell and present them on MHC class 1. And if you block that because you're a virus, then you can prevent the presentation of that antigen and thus keep your cell alive at least longer. So the next family I want to talk about are interleukins. So interleukins are a super diverse set of proteins. They have subfamilies too that I'm not going to go into, but basically what they bind to and trigger is usually associated with leukocytes, which is our generic term for white blood cells. So if you hear leukocyte, that's just an older school name of white blood cells. They're incredibly diverse. It depends on the organism that you're in, but they're generally only associated with leukocytes. I mean, I say that, there's of course exceptions, but they're just interleukins. So leukins being like a white blood cell protein and inter being in between. And so it's generally just a way for white blood cells to talk to each other. So if you look at this chart, and I have a better chart on the next page too, you can see a lot of the signaling patterns are actually shared between the receptors of interleukins as well as other cytokines. And so we've got lots of JAKs and lots of STATs that you'll see on here. So the JAK-STAT pathway as well as the MIghty88 pathway are really, really common pathways that get triggered and then eventually go into the nucleus as transcription factors. And then you just pick which stat and which JAK you want to use, and then the JAKs basically end up phosphorylating the stats, and then you decide which stats I want to use, and those stats will decide, okay, what gene do I actually want to amplify? And so if we jump over here, you can see there's a lot going on here. I understand that. This isn't meant to be comprehensive. It's just to show that there's a lot of complexity and the actual presence of specific cytokines. In this case, we're looking at interleukins and mostly interferon. There's some TGFs and TNFs that we'll talk about in a minute. But you can look over here. So we get the binding of interleukin 12 and interleukin 15 to these receptors. Those stimulate these specific JAKs, which stimulate these specific STATs, which stimulate these specific genes, which then cause these different effects. So this chart is actually really, really cool and really helpful. And so you can see some of these things that are really interesting. So interleukin like 12 is associated with like cytotoxic T cells. Interleukin 10 is a great example of an immunosuppressive cytokine. So interleukin 10 would be associated with like T regulatory cells and B regulatory cells. It's the quintessential cytokine secreted by those. Whereas other cytokines are more associated with like inflammation. So interleukin 17 would be a great example of an inflammatory one. It's not in this chart, but that would be a really powerful one that would cause inflammation. Interleukin 1 as well, interleukin 1 beta would be a great example of an inflammatory cytokine. The next family I wanted to talk about is actually super diverse, and it actually covers basically all of these. And so we've talked about hematopoiesis before, so that's how an initial stem cell develops in the bone marrow into all the different white blood cells. And so the different cytokines that are present can change what that cell actually becomes. And so this isn't an exclusive set, so there's not like hematopoietin 1 and hematopoietin 2. We actually see lots of the other ones. So you can see lots of interleukins here. You can see lots of growth factors, granulocyte, monocyte, colony stimulating factor. So all kinds of just different things can come in here, but we consider them hematopoietins if they're influencing the development of cells, and a lot of them do. So the next family I want to talk about are the tumor necrosis factor family. So in the late 60s, we took tumor necrosis factor and we put it on some cancer cell lines and they killed them. So they called it, oh my gosh, tumor, cancer, necrosis, death, factor. It's something that's killing these cells. And they're generally pro-inflammatory cytokines. And so you can see a lot of the effects that they have are generally going to be inflammatory. So they're going to increase like interleukin 1 beta secretion by certain cells, like macrophages basically. They're going to keep immune cells alive because we tend to associate inflammation with immune cell activity. Now that's opposed to the transforming growth factor beta family, which is kind of the opposite. So the TGF-beta family is generally immunosuppressive. And so a lot of the studies of TGF-beta come from cancer cells. And so a lot of cancers will secrete TGF-beta. It doesn't have to be exclusive to cancers, but they will secrete TGF-beta, and that will suppress the immune system. And so TGF-betas tend to be immunosuppressive. That doesn't necessarily mean that they are anti-inflammatory, but it's kind of hard to tease out and separate the difference between the immune system and inflammation because they're so closely tied. And so if you can suppress inflammation, you're generally suppressing the immune system. TGF-beta has a lot of different effects, but they actually kind of make sense. So I like this chart right here. So you can see, so CTL is cytotoxic T lymphocytes. So those are your CD8s, your T killer cells. So you can see TGF-beta tends to suppress them as well as natural killer cells. And those are the two big cell types that are going to be attacking cancer. So that makes perfect sense that cancer would secrete TGF-beta because it's blocking these cell types that are going to go and kill the cancer. We also see a blockage of like your dendritic cells because you're trying to keep them from presenting tumor antigens. And we also see an increase in T regulatory cells. And so those are the T cells, the T helper cells that are associated with the secretion of interleukin 10, which is an immunosuppressive cytokine. And so that makes perfect sense, though, because you would want to increase your T reg presence and what they're doing in order to suppress inflammation and the damage that it could cause to like a cancer cell. The chart I have over here is going into way more depth about the actual transcription factors and where they bind. And you can see it gets really complicated really fast. But basically, you've got your tumor necrosis factors, which are pro-inflammatory, and you've got your TGF-betas, which are anti-inflammatory. So I've hit basically all the families except chemokines, and what I wanted to talk about just for a second is that a lot of these share similar pathways. And so you can see you can get these growth factors or your cytokines or whatever because we can kind of use those interchangeably. And they have a lot of the same signaling pathways. So a lot of RAS and RAF, a lot of kinase activity, a lot of STAT and a lot of JAK-STAT pathways. And so there's some crossover happening. And so you can actually see some of these pathways can influence each other too. So the I kappa B can influence the NF kappa B, which could be triggered by other survival factors, but that actually came from cytokines. And so it gets really complicated really fast. But basically, all of these things can really have a lot of different effects on each other. So that leads us to our last family that we're going to talk about, which are chemokines. So chemokine stands for chemotactic cytokine. And so it's a chemical taxis, i.e. movement, using cytokines. And it's basically just an attraction. So what chemokines do is you have your ligand and your receptor. And so a tissue that wants to attract a specific cell type will secrete a specific chemokine ligand. And then a cell on the other end would express a specific chemokine receptor that can bind to that ligand. And it basically uses that like a very specific nose.
Oh, Stewie and Brian, you're just in time for
Chemokines.
Did somebody say
Chemokines? And so that nose is sniffing for specific chemokines. And if it smells the most delicious one that binds to it, then it gets attracted. And as your concentration gradient increases of that ligand, it causes the cell to move in that direction. We call that chemotaxis. So chemotaxis is just the idea of either moving towards something, that would be a chemoattractant, or the idea of moving away from something, which would be a chemorepellent. In general, like bacteria do this, and so that's why it's talking about attractants and repellents. In general, chemokines are just chemoattractants. So what's kind of cool is you can look at specific tissues and they kind of make sense. So one of the better examples would be, let's say your macrophage gobbles up a bad guy and it heads to the nearest lymph node. Well, once it actually enters that lymph node, what do macrophages want to do, or other antigen-presenting cells like dendritic cells? Well, they want to present that antigen to as many T helper cells as they can in the hopes that they can find a T cell receptor that matches, right? And so they're going to attract T helper cells, and they're going to also attract B cells to the lymph node in the hopes that they can get that connection between a macrophage and a T helper cell and a B cell to actually trigger an immune response. That whole process is triggered by the secretion of chemokine ligands from the immune cell, or from the macrophage, the APC, and binding to chemokine receptors on other cells. So another example might be, let's say we have a T helper 1 inflammation, so that's generally caused by a viral infection, and so that tissue is specifically secreting CXCL9 as its ligand and CXCL10 as its ligand, and those are going to bind to CXCR3, which is a receptor, and it's going to attract T killer cells to that site, which makes perfect sense because if you have a viral infection, you want T killer cells attracted to the site of the infection, so that makes sense. So you'll notice all of our chemokines, they all have CC something or CXC something. And then they have L or R for ligand or receptor. And so what's happening is they share a commonality structure. And so you can see the C's stand for the amino acid cysteine, which that's the code for cysteine, right? And so you can see here, all of these chemokines share this similar structure. And so we're kind of avoiding the C chemokines, but so if you have a CC chemokine, then you've got two cysteines next to each other. If you have a random amino acid in between those cysteines, you still have those cysteines. They're helping hold that secondary or tertiary structure of the chemokine together. But if you have an amino acid in between them, then we call it a CXC chemokine. Or you can have a CX3 chemokine. But what's really interesting is you always have this consistent set of cysteines that form this specific structure. And so you see commonalities between chemokine ligand structures as well as chemokine receptor structures. The signaling pathway of chemokines is really similar to all kinds of other pathways. And so over here, we've got our chemokine receptor, and here we've got our chemokine that was secreted by something. And then in this case, it was a paper talking about something else where like another cell would express this, which can sort of help the cell be drawn into a tissue, which we're going to talk about that in a minute. But basically, you can see this chemokine is able to, based on its residues here, which has to do with the actual structure of these cysteines, it's going to be able to fit onto the specific residues here and bind to it and trigger it. Something interesting to note, lots of 7 transmembrane receptors exist. That's just a really common motif that we see. So we're talking about chemokines. Immune cells in general don't necessarily have to go just towards chemokines, though that's kind of what we're talking about right now. They could be chemoattracted to all kinds of things. And so I had a video here that they're showing of neutrophils being attracted to a really powerful antigen that they would basically want to attack. And so they're trying to study, okay, like if I space out my neutrophil, how close do I actually have to get this thing that the neutrophil would normally want to kill before the neutrophil starts acting weird? Which is pretty cool. You can see that neutrophil, it just starts freaking out and like reaching out for that thing. So creepy, but kind of awesome too. And so when neutrophils sort of smell something bad, or if they're expressing a chemokine receptor and they smell a chemokine ligand, they're chemoattracted to a specific tissue. We call that extravasation or diapedesis, which I know both of those are huge words, especially diapedesis. I never liked that term. I thought it was kind of silly. Extravasation makes more sense to me because it's extra, meaning like out, and then it's leaving out of a vessel. So it's like vas or vasation. I don't know. So I just like extravasation better. That's generally what I call it. But diapedesis sounds cool too. And what you're actually seeing is the movement of either a monocyte or a neutrophil or some kind of circulating blood cell that needs to get into a tissue. And how does it do that? Well, that's a great question. It's really, really important. We call that paracellular passage. It could also be transcellular, where it goes like through a cell, like literally the endothelial cell will allow an immune cell to like pass through it, not through the sides of it, like around it, but like literally through it. That's super weird. But generally, you're going through the paracellular space in between the endothelial cells. And how this actually happens is really, really interesting. So rolling along, and your neutrophil is expressing a specific set of proteins, and the endothelial cells are also expressing a specific set of proteins. And all of these things are triggered by inflammatory markers. So you kind of assume, okay, down here, I have like a site of infection. So I have bacteria or something, and my resident macrophages or neutrophils or whatever are freaking out because there's a bacterial infection. And so they start secreting cytokines that trigger changes. Okay, so they're secreting chemokines to attract things to the site. They're secreting cytokines to trigger specific types of immune cells and immune cell maturation, like for hematopoiesis. And they're also triggering the changes that would allow a cell to enter into a tissue. And so in this case, your neutrophil or monocyte or whatever it is are going to express a variety of things on their surface, a variety of proteins that can grab onto a different set of proteins on the endothelial cells. And that allows the cells to then enter into the tissue. And I have a nice, cool video here that I'm going to show.
Extravasation of neutrophils. White blood cells circulate in the blood, searching for pathogens within our bodies. Neutrophils, the most abundant type of white blood cell, patrol the blood for signs of infection. When inflammation is detected, neutrophils become sticky and roll along the capillary wall, adhering to it and eventually squeezing out of the vessel. This entire process is called extravasation. Extravasation begins when chemokines are released by immune cells at a site of injury or inflammation, activating the endothelial cells of nearby capillaries to express adhesion molecules. Neutrophils respond to the adhesion molecules by expressing counter receptors, which produce a weak interaction between the neutrophil and endothelial wall. The neutrophil adheres slightly to the vessel wall, rolling along the surface. This movement is possible through unique molecular interactions between neutrophils and the endothelial cells. The neutrophils are slowed down by the formation of weak transient bonds between Sial-Lewis X, a carbohydrate expressed by neutrophils, and selectins, adhesion molecules produced by endothelial cells. The force of blood flow causes neutrophils to roll along the endothelium, causing new interactions to form while old ones break. Eventually, tight binding occurs when integrins known as LFA1 on the neutrophils undergo a conformational change, causing them to bind firmly with endothelial adhesion molecules called ICAM1. With the neutrophil firmly adhered, diapedesis, the process of neutrophils exiting between endothelial cells, can begin. Once outside the blood vessel, the neutrophil migrates to the infection, guided by chemical signals.
So that's a really cool video. You can actually find a lot of cool science videos. They were like master's projects from science artists or science communication specialists. So that's kind of fun. And the very last thing I wanted to talk about, something you may have heard of before, is called a cytokine storm. And so if we're talking about cytokines and chemokines and all this stuff, well, what's been in the news lately is that COVID can actually cause a cytokine storm, and they think that may be responsible for what's killing some of the people that get COVID. So we need to talk about cytokine storms because it's something that you might encounter at some point. So a cytokine storm is also called cytokine release syndrome, and basically your body has entered an inflammatory state and it's not able to put the brakes on. So you're going to get the release of tons of inflammatory cytokines, so your tumor necrosis factors and your interleukin 1s, those are really bad. So you can see here, like there's tumor necrosis factor alpha and interleukin 1 beta. Those will cause inflammation like nobody's business. And if you don't control that, then that would be bad because you're kind of always balancing that. Do I need to cause inflammation, which is going to help me clear this infection? Or is too much inflammation going to actually damage the body? And a lot of times that's what's actually killing people is there's just too much inflammation. What can actually cause this? Well, that's a good question. Generally, there's super antigens, and so that's something we haven't talked about before. A super antigen is something that indiscriminately will bind to T cell receptors and trigger the activation of a T cell without an APC's help, which is terrifying. And so it's triggering not just inflammation, but it can also trigger autoimmune responses because, as we've discussed last time, your T cells are supposed to be deleted if they attack self, but that's not a perfect process. And so if you're accidentally triggering a bunch of T cells, so let's say like 10% of the T cells that get out of the thymus are actually self-reactive and we have to keep those suppressed. Well, I'm indiscriminately activating them with super antigens that are just binding to their T cell receptors randomly and triggering activation without an APC. And so what happens is self-reactive T cells can activate. Those can activate self-reactive B cells. Now you have auto activity, so you're attacking yourself, so you have autoimmunity. And it can also trigger just the release of inflammation, or inflammatory cytokines, which is really, really bad, obviously. You can trigger this as well in certain medical situations. So like if you throw in like an antibody that could grab onto it and basically bind to and force dimerization of the T cell receptor, then you could accidentally trigger this whole process too. And so we see that in certain treatments because you can use antibodies as treatments. So if you have a patient, especially like a COVID patient or somebody with some kind of infection, you're going to want to do a full panel and look at their actual cytokines. And then you're going to look at the levels of different cytokines. You're going to say, I see a lot of interleukin-1 beta. I'm seeing a lot of TNF alpha. And I probably should put them on a steroid now because that's going to be one of the best ways to keep that immune system suppressed. You could also do things like put in an antibody that blocks specific ones. So like an anti-interleukin-1 antibody might actually be effective too. But in general, you just want to suppress that inflammatory response. And that, my friends, is cytokines.